All right, if you're listening, then I know that you're interested in data, so I am too. And let me show you the data that we have that seems to suggest that cross-reactivity is important to immunogenicity. If something is cross-reactive, it is more likely to provoke an immune response, whether in a previous infected or even an uninfected person. Remember that the infections we're talking about can be common cold infections on the one hand, provoking SARS-CoV-2, or they can be SARS-CoV-2 actually going backwards and reacting to a common cold peptide, which is in one of the later experiments. So remember again that with the grain of salt that we only looked at one class 1 HLA, and we know that everyone has several. A population has many more. We looked at the most common ones, but even the most common one is only in 25% of the possible genes that you have. And so we know these populations are more complex. We're simplifying, and yet we still see significant response for the cross-reactive peptides. They're significantly enriched. When you look at an experimental peptide and you see a result, then you go back and you see this activates T cells. Then you go back and see, I wonder if it's homologous to a coronavirus common cold. More often than not, you see that there's something there that is suggesting cross-reactivity is important. Okay, so we have a couple of different things. I actually had a typo in the previous one. We have 4 different studies that I'm going to quickly show you their experimental data. And of course, I'm going to let you have the fun of going in and finding the studies. These have also been previously referenced, and so sometimes you might have to, you should be able to find them just from the specifics that I say right here, but we have previous ones if you have any trouble finding them, and they're on the previous slides. Okay. So Grifonia Dial, the one that came out in May, that came out in Cell in June, I believe, had these data on the bottom. Remember that had the data for unexposed patients that were reacting to class 1 and class 2 T cells. I put that data on a bar graph. Every time that they had a patient that was above the line showing that they had some kind of T cells that were reacting to this, even though they had never been infected with SARS-CoV-2, I put a bar on the graph. And so you see they only had 20 patients. And in fact, I think they only had, if I remember correctly, they only had 10 patients in each. So that means that this may be 10 or 20 patients. Out of those, yeah, I believe it's 10. Then you have these different patients had T cells that reacted to these different proteins from the SARS-CoV-2 virus. And so separated out by genes, because that's the way that they did it. They separated out by peptide pools. So if you see on the bottom there, you tend to have, there's only a couple of places where you don't have cross-reactivity. What's interesting to me is out of this pool, when we have cross-reactivity goes together with unexposed T-cell reactivity 15 out of 17 times, out of the 17 times that we're showing right here. There's only 2 cases where you have cross-reactivity where you don't see unexposed patients having some T cells. You can see that the shapes of these bar curves are roughly the same. Especially look at the structural proteins all the way on the left. They're the ones with the single letters. M, N, S, and E. M and S have a lot of cross-reactivity, and lo and behold, when you go to the bottom, they have a lot of T cells associated with them as well. N and E don't have a lot of cross-reactivity. There's only one, and that goes below the threshold of what is significant cross-reactivity for this. N and E don't show any unexposed patients reacting to them. This seems to say that the cross-reactivity may play a role in priming T cells for a reaction against something that they've never seen before. So that was class 1. Grifonia Dial also looked at class 2. Here's the same data for class 2, and you'll notice this data is not quite as clean. The shapes of the bar graphs are not exactly the same, but we still have the tendencies for 11 out of 17 of them. When you have cross-reactivity, you also have unexposed patients reacting, and vice versa. You had to have both cross-reactivity and unexposed, or neither. Both or neither counted as a positive 11 of 17 times. And you can see that, again, we're probably running up against the low amount of the study. How much of this is experimental noise? How much of this is something different in how class 2 peptides actually work? But again, if you look at the structural proteins, you get a clear picture where you have cross-reactivity coinciding with T-cell reactivity in the case of unexposed patients. Here's something that just came out. It came out for the IEDB database, and this is a paper that's a preprint right now. It comes from Adaptive Biotechnologies, which is a Seattle hometown company. And what they do is they scan for class 1 epitopes. They took a bunch of patients. Some of them were even actively infected. Most of them were post-infection. So these are infected patients, not non-exposed like the previous one was. 248 class 1 epitopes are what they found in the regions of the viral genome that we looked at. So we put our predictions on top for cross-reactivity, and then we look at the bottom to see how many of those epitopes were found in their search for different class 1 epitopes. Again, this is experimental confirmation of the same epitopes on top. So you're not going to get a number that's greater than 100%. You're only going to be creeping up to 100%. So the bottom graph is always going to be smaller because 248 epitopes rather than, what was the number, 790 some epitopes. They only found 248 epitopes out of our, and there are way more than that in the SARS-CoV-2. So all that is to say, you expect the lower graph to be smaller. And as I'm talking this out, I'm looking forward to actually writing this down because that's the way I'll really be able to explain it better. But the important thing is we looked for those 248 epitopes that we know exist in infected patients. How many of them cross-react with flu, or with common cold? The answer is 147, or 58% of those epitopes are cross-reactive. Compare that to the whole SARS-CoV-2 epitope pool, for which 1/3 of the epitopes cross-react. And you'll see more of these experimentally determined epitopes are cross-reactive than you would expect if they were just drawn at random from that pool. You'd expect them to be about 1/3 reactive. Instead, they're about 60% reactive. So it looks like cross-reactivity may double an epitope's chances of being immunogenic. Roughly speaking, back-of-the-envelope calculation here. And in fact, the one thing I noted for all of this, when you look all the way over at the structural proteins, those bars are almost the same on the top and the bottom. That means that all of the cross-reactive epitopes were found by Adaptive when they were looking for epitopes. All the epitopes, now notice the way that I'm saying this is very restrictive. Of the cross-reactive epitopes, of the epitopes that were cross-reactive, 26 out of 28 of those were found experimentally by Adaptive. Okay, so the students predicted that they would be cross-reactive, and we found out that they were cross-reactive. I break it down by the different viral strains just to show you this cross-reactivity is a general feature. There's not many places where you see fewer than 4 colors. So for all of these, we're likely getting a contribution from all 4 of the viruses to the pool. So could a common cold basically prime T cells to be ready to react? Could it be the memory T cells that are originally activated by common cold coronaviruses are the ones that tend to be reactivated when challenged with SARS-CoV-2? And so that is the other experiment. The other one that I didn't show that I have on the slide is actually the one that we started. I showed it before when we determined the class 2 thresholds. The class 2 thresholds were determined from, I have to look at the name, Mateus et al. And they actually, we're actually looking at the cross-reactive peptides. And so we're kind of taking the thresholds from them, and then we turn around and apply them to just our HLA. With that said, again, we're comparing a situation with just our HLA to an entire population of HLAs. and yet we still see 5 of the 9 peptides cross-react will work with our HLA, our one little HLA. I don't have the figure for that one because I already cited it before, and it doesn't really show you that much. Okay. Now, the last thing that we can say is I've shown these data before. This is from Chauer et al., and this is where they looked at just 2 people who had been infected and 1 healthy donor. But they identified with the dotted lines that you can see, 5 dotted lines, they identified 5 specific peptides that they found both reacting a lot in the infected people on top and infecting a little in the green bars, the healthy control on the bottom. By the way, the Y scale is different. That's why the bars look about the same size. They identified these from an experimental screen as immunogenic peptides. 4 of the 5 of them were found also to be cross-reactive. We didn't have any knowledge of that. We didn't put it in, and we just happened to find it that 80% of the peptides that they identified, we also identified as cross-reactive. And that's why I'm starting to say I think there might be some causation here. By the way, the one far on the left doesn't match any common cold viruses. There is a theory, I believe it's Mateus et al., that has a theory that it might match animal beta coronaviruses. So it's possible that you could get a little infected with an animal coronavirus, never know it, and it could give you a little bit of immunity maybe to that epitope. It's a likely theory, but of course it has to be tested. But all this is to say, 5 lines, 4 of them would be predicted by just looking and seeing what overlaps with a common cold. So that's our experimental data so far. I think this is stronger than I thought it would be. I wasn't sure that I should write it up, and now I'm looking at it and talking it through, and I'm thinking I should write it up. So I don't know if you happen to be working on this and you're an immunologist who'd be interested in taking this data and using it in any way, definitely contact me. But thanks for at least giving me the opportunity to explain this. And all my students who are listening, look what your work has done. You know, you can at least show people what this video is. They don't have to listen to it all, but they can sort of page through and see the different slides that show you what you've accomplished. We have found that the cross-reactive peptides that cross-react with common cold and SARS-CoV-2, also appear to be experimentally immunogenic. And like I said, we can't say cause or effect, but I'm pretty suspicious. So let me know what you're thinking. I'll keep my students, I will keep you up on what goes with it from here. And until then, take care, everyone. Take care of your T cells.